have taken the spotters which I have completed their syllabus units of carbohydrates, proteins, amino acid and enzymes. So these students are very much eager to share their given spotters to you in this video. So let's start the video.
Hi, this is Sajida Fatima from the Department of BSc Cardiac Technology. So now I've been given a spotter that shows two different structures of carbohydrates and I'll be identifying them on what each structure are. So here, coming to the first structure, for the identification we consider the functional group. So now we have KO, which is ketone functional group. So this is a keto structure. And for the second step of identification, we consider the number of carbon atoms. So we have 1, 2, 3, 4, 5 and 6 carbon atoms. So this is a hexostructure. And for the final step of identification, we consider OH based on which side it is located. Based on which side it is attached to the chiral carbon. So OH is attached to the right side means this is a dextrorotatory structure. So summing up all the identifications, I identify this as fructose structure. So this is a dextrorotatory keto hexostructure. Similarly, for the second structure, let's consider the functional group which is CHO, that is aldehyde. So this is an aldose structure. Secondly, we consider the number of carbon atoms where we have 1, 2, 3, 4, 5 and 6 carbon atoms. So this is a hexostructure. And similarly, we consider OH based on which side it is located and attached to the chiral carbon. So it is located on the right side, means this also is an dextrorotatory structure. So based on all the identification, this is identified as glucose. It is a dextrorotatory aldohexostructure.
Hello, I am Varsana from the Department of Cardiac Technology. In this video, I am going to explain about a spotter on protein structure. This spotter shows different level of protein structure: primary structure, secondary structure, tertiary structure, and quaternary structure. This is the primary structure. It refers the linear sequence of amino acids and it is formed by peptide bonds. This is the secondary structure. This is formed by local folding of polypeptide chains. There are two types of secondary structure, alpha helix structure and beta filter shape. The alpha helix structure is in the this is the tertiary structure and it is formed by three-dimensional structure of peptide bonds. And it is stabilized by hydrogen bond, hydrophobic, ionic and disulfide. This is the quaternary structure and it is formed by multiple polypeptide subunits. And this is the quaternary structure and it is formed by multiple polypeptide subunits and it has an example of hemoglobin.
Thank you everyone, I am Vinodini from the Department of Cardiac Technology. Today I am going to explain about the given spotters forming lipid pigmentation. Lipids are organic compound insoluble in water and soluble in organic compounds like ester, chloroform. This is green capsicum. Chlorophyll is present in green capsicum. In red capsicum, lycopene is present which gives red color. In yellow capsicum, xanthophyll is present which gives yellow color. In orange capsicum, carotenoids is present which gives orange color. In tomatoes, lycopene is present which gives red color and ripe tomatoes. In carrot, beta carotenoids is present which gives orange color. It is rich for vitamin A. In watermelon, lycopene is present which gives red color and ripe watermelons.
Hello everyone, I am Aishwarya from BSc Cardiac Technology. Today I am going to explain on a given spotter for me which is given as enzyme. Enzyme is a globular protein. Enzyme which boosts up the speed of reaction in the living organism, act as a biocatalyst, and it also absorbs nutrients, helps in excretion, secretion, reproduction, and also function of kidney and liver. And it has a 3D structure. It is mainly classified based on its structure, which is primary, secondary, tertiary, and quaternary structure. And the enzyme has an active site, where the active site is a small region where the substrate come and bind the active site and the reaction occurs. And the substrate is further formed into enzyme substrate complex and it is enzyme reaction.
So far our students have clearly explained their spotters and this is the carbohydrate structure of fructose and glucose, where the functional group will get vary from one another, and fructose having ketone functional group and glucose having aldehyde functional group, and Shajita have explained it clearly. And second spotter is about the protein structure where proteins have 4 different structures of primary, secondary, tertiary, quaternary, and Shajita have explained clear about the 4 types of structure of protein and its examples also. And third one is all about the lipid classification, which comes under the derived lipids, and this is all about the pigments which is involved in the fruits and vegetables. And Vinodini have explained clearly about the pigments of capsicum, tomato, and carrot, beta carotene, and the watermelon, and these pigments are responsible for having its antioxidant property and its importance in health also. And finally, this is about the enzyme structure, substrate, and the enzyme is a globular protein where it binds with the substrate, and this bind with the help of active site region, and this is the site where all the substrate will bind into the enzyme, and the enzyme substrate combines to form enzyme substrate complex, and after forming the complex the product get released and the enzymes also released for binding with the another substrate. So these are all about the biochemistry spotters, and thank you so much students for involving and participating in this.